All right, Lab 10. That means we are almost done with this semester. There's only a couple left, so woo! So this lab, you are going to be building on genetics information that you've been talking about in lecture, and we are going to be using some math, probability, and statistics in order to confirm if hypotheses of certain genetic matches are actually confirmed or denied. So yes, we're going to have to pull out a little bit of math, but before we do that, we are going to need to talk a little bit about the background of genetics. So again, this should be building on material that you've been looking at in lecture, but just so you know what's expected of you here in the lab, make sure you go through your objectives and you should be able to answer and explain all of these when you are finishing up the lab. And of course, your keywords, which you should be able to spell and define at the end of this lab. So we're going to spend some time talking about genetics. What is genetics? What are some of the terminologies that you will need to know? And then, of course, we will jump into the math. That's the fun part. Also note that in YouTube, you have some math full explanations. So if that's something that's making you a little nervous, make sure that you look at those as well. So let's talk a little bit about genetics. So genetics is looking at how things are passed down from generation to generation. Now this all started in an abbey with a monk playing with peas. Again, something else you'll talk a little bit more about in lecture. So back in the mid 1800s, Gregor Mendel was breeding peas, and I mean like garden peas, like the green peas that you eat. And those peas were able to basically start our entire study of genetics. It's absolutely amazing. So by crossing certain types of peas, for example, a tall plant with a short plant, or a plant with white flowers with a plant with purple flowers, he was able to trace and track what happened from generation to generation. And this is kind of that mentality behind what you often hear in layman's terms as things skipping a generation. We're going to talk a little bit more about how that works here in this chapter. And there's a beautiful picture of Gregor Mendel. Fancy. So, a little bit of terminology that we need to look at. So, the first thing is to understand the difference between a character and a trait. So, a character is something that is going to be a little different from organism to organism. So, one of the great examples of this is when we talk about flowers, right? We're looking at plants from the Abbey Garden. So, when Gregor Mendel was looking at peas, he was looking at many different characters of that plant, how tall it was, what its color of the flower was, what its seeds looked like. So let's take, for example, the flower. So the character is the fact that the plant had a flower and it had a color. The trait is the type of that character. So what color was that flower? Again, with the example that Gregor Mendel used, he had both white flowers and purple flowers. So those are the trait. So the white flower is a trait. It is a type of the character. And again, you can see here in this image all of the different things that Gregor Mendel was looking at. So he also looked at the color of the seed and the texture of the seed. So the color of the seed is the character, and whether it was yellow or green is the trait. So be careful as you are reading and as you are explaining what you are seeing as we go through this lab. So the other thing that we need to talk a little bit about is some terminology that you're going to hear quite a bit that might be a little bit different than what you hear in layman's terms, right? When we're just talking to, our friends, we tend to use these terms a little bit differently. So when we talk about genetics and we look at what Mendel was doing and the examples we're going to use in lab, we're going to use two words, dominant and recessive, to explain when traits are seen and maybe when they're not. So we're looking at a dominant allele, which you talked about when you did your mitosis and meiosis lab, yes, or a recessive allele. So the allele is the gene that gives us the trait, right? So when we talk about a dominant trait, it is the one that will be, if the genetics are there, it will show up. The recessive, on the other hand, is going to be masked. It is going to be covered up if there's only one. If there is a dominant allele present, the recessive allele will not show up. So then two other things to think about in this lab is the difference between phenotype and genotype. So phenotype is physical, so the alliteration there. Phenotype is physical. The physical feature is what we can actually look at and see, what we can note. So like the color of the flower is a phenotype. And we can also figure out what is the dominant phenotype versus the recessive phenotype. Genotype, on the other hand, is a little bit more challenging, right? Genotype is looking at the actual alleles. What is your combination? So in your meiosis lab, you learned about alleles and you talked about how everyone has two of the alleles, one from each parent, right? So with the genotype, we can talk about this in three different variations. So we can be homozygous. Homozygous means we have two of the same alleles, meaning we either got two of the dominant alleles or we got two of the recessive. So if you got two of the dominant alleles, you are considered homozygous dominant. You have two of those dominant alleles. Now, when we start writing these, we will often use letters to notate this, right? So homozygous dominant would be capital letters. So I always use R's, which is the example I'll actually be using in a little bit. So I would call someone who's homozygous dominant big R, big R. Now, you can also be homozygous and be recessive, which means you have two recessive alleles. So again, as we write these, we use letters. These would be your two little letters, little r, if you will. And you can use whatever letter you want. Just make sure when we're talking about the same character, you're using the same letter. So a homozygous recessive should not be notated as like little r, little a, right? Make sure you're using the same two letters so that you can stay consistent. Because when you start talking about other characters, we tend to use different letters. So stick with one letter when you're talking about one character. Now then the other option we have is to be heterozygote, which means we have two of each kind. So we have one dominant gene and then one recessive gene. And that means that we would use a big R and a little r. So what we're going to be doing is using these genotypes to hypothesize what our generations would be if we were to mate these together. So let's talk a little bit about what Mendel did in order to kind of figure out all of this. Because again, he didn't have molecular biology like we do. He didn't have DNA testing. He really just had the mating and following certain patterns. So in his garden, he was able to look at one single character at a time by really carefully mating them together. So the simplest of the crosses that's able to be looked at is called a monohybrid cross. Mono meaning one, where we're looking at one single character. Now again, I think Mendel got kind of lucky where he picked characters and he picked a plant that only has two traits. So when he looked at height, or in this case, in this image, you can see he looked at colors of the flowers, there were only two options. It was either purple or white. There weren't a whole bunch of different types. So when we talk about a monohybrid cross, we are looking at one single character that has two options, two traits. So what Mendel did was he took two what are called purebred organisms, meaning that they had purple flowers no matter what all the time. If they mated, they made purple flowers, purple flowers, purple flowers. And then another plant that was also purebred that had white flowers only, white flowers, or if they mated, that's all that they would get. And he crossed them together. Now these are called the P generation, the parent generation. Basically, these were both homozygous. The violet flowers were homozygous for the purple, and the white flowers were homozygous for the white, and mated them together. And what came out was not something that he was expecting to actually happen. This next generation, which is called the F1 generation, was all purple flowers, right? So all of the plants came out purple. There were no whites at all when these two were mated together. Now, this is where that concept in layman's terms of skipping a generation kind of came into play, because everything was purple. But when he allowed for these plants to self-pollinate, meaning that the egg from the purple plant was able to be fertilized by the pollen from the purple flowered plant, the next generation did see these white flowers appear. Now, when he counted it all up, what he found is 3/4 of these flowers were purple and 1/4 were white. So this gives us what we now think about as a 3-to-1 ratio. For every 4 plants, 3 of them come up with the dominant trait, which is purple, and the 1/4, 1 out of the 4, comes out with the recessive phenotype, which is the white. So we started with a dominant phenotype mating with a recessive phenotype, and we had all dominant babies. Then we allowed those to mate with themselves, essentially, like they were self-fertilizing, self-pollinating. And we created some dominant and some recessive in a 3-to-1 ratio. Now we can use this information to hypothesize what should happen as we move into other experiments, which means we've got to talk a little bit about the Punnett square. Now many of you are familiar with the Punnett square, but I do want to walk through this with you to kind of show you how the Punnett square is going to give us a hypothesis. And again, remember, that's all we're doing with the Punnett square is creating a hypothesis. So we can predict what may happen if 2 individuals mate with each other. Now, one thing to remember here when we create the Punnett square is, as we learned in meiosis, when every person, every flower, every organism has 2 alleles, they will separate on the homologous chromosomes during meiosis, right? So one allele will go into one of the gametes and the other allele will go into the other. So that's how we create this Punnett square. So let's look at our example here. Parent 1 is homozygous dominant, which is going to be written as big R in my example. Parent number 2 then is purebred for the homozygous recessive with the white petals, and I will write the parent 2 as little r. So in order to create our Punnett square, we're going to draw a little box. I basically think about it as a tic-tac-toe box. Now I made it a little fancier on the screen because I can, but it's basically a tic-tac-toe box. We are going to fill in the top row and the far left row with the parents' alleles. So each box is going to represent a parent's gamete. Now, we're only going to look at one of these genes, so we are going to put just big R representing the dominant allele in parent 1 across the top. And we will put parent 2 along the sides. So these 2 little r's are each in one box representing the gamete of parent 2. And that's how you set this up. So you spread them out. When we're only looking at one character, we do not need more than one letter in these top 2 boxes. But remember, every baby is going to need 2 genes. So we're going to put these together. So in these 4 little boxes here, each one of these is going to represent one child. And again, this is a hypothesis. This is not always going to happen, but it gives us a good hypothesis. So in order to create the Punnett square, the first thing we need to do is bring down parent 1's gene. So the big R is going to go into each box. So every child in this situation is going to get one dominant allele with the big R. Then we can bring over parent 2's gametes to each kid. So again, in this case, every single child is going to get a recessive gene from parent 2, which means in this situation, all of the children end up the same. So our hypothesis is that all of the children are going to end up as heterozygotes as a big R, little r. Now, as you do different types of Punnett squares, you will find that this is not always the case, but this is the example that I want to talk about. Now, the other thing that you will potentially see as we go through Punnett squares is how to create ratios. So the ratio of the phenotype is what each individual looks like. So remember, a homozygous dominant and a heterozygote will both appear dominant. They will have the purple color, if that's the example that we're going with here. So in this case, all of the children ended up purple because they all have a dominant allele. How many of them ended up white? None. None of them are little r. None of the children in this case are little r. So our ratio is a 4-to-0 ratio. Now the other ratio that I could ask you about is genotype ratio. So the genotype has to look at all 3 of the different types of genotype. First, homozygous dominant. We have none here, right? There are no homozygous dominant individuals that were created or that could be created in this cross. Now, there are 4 heterozygotes that could be created in this cross, but there were none of the homozygous recessives. Now we do have to include all three numbers here, even though it is a 0. You can make this a more simple number if you want to, but you don't have to. So a 0 to 4 to 0 ratio would be our genotype. So again, we're comparing the genetics with genotype to our phenotype and what we can actually physically see.